Friends, welcome to Biology Made Easy. Today in this video, we'll discuss the duplicate genes or pseudo-alleles which are coming in the ratio of 15:1. That means Mendel's dihybrid ratio 9:3:3:1 is modified to 15:1. So in this case, in case of duplicate genes, what happens? There are two independent genes present in different loci, but their behavior is same. That means individually they produce a particular trait, and when both of them are present together, the same trait is produced. Suppose this is a chromosome. In this chromosome, suppose here the gene A is there, either in the dominant form or recessive form, and here gene B is there. That means they are present in different loci. So usually what happens, both capital A and small a, they produce the same character. Like we know in Mendelianism, suppose capital T or small t, both of them are responsible for the height of the plant body, either tall or dwarf. Similarly, the round or wrinkled, they are responsible for the shape of the seed. So similarly, here capital A and small a, they will produce a particular character, suppose it is X. And here B, either capital B or small b, that will also produce the same character X. That means both are duplicate of each other. And again, when both A and B are presented together, they also will produce the same character X. So this is the difference between this Mendelian behavior and this is non-Mendelian behavior. And why it is known as pseudo-alleles? Pseudo means false. Usually the two alleles of a particular gene produce a particular character. But here the genes are present at a different loci, but they produce the same character. That is why they behave as pseudo-alleles. That means they behave just like alleles, but they are not true alleles. They are pseudo-alleles. That is why duplicate genes are also known as pseudo-alleles. So let us give examples. So one of the very interesting example for this duplicate genes is given by G.S. Searle, or you can tell George Searle, in case of shepherd's purse or Capsula bursa pastoris. So George Searle gave the example from shepherd's purse. Shepherd's purse, and it is the botanical name is Capsula bursa pastoris. This is the botanical name. So in this particular plant, he studied the fruit shape. And the fruit shapes are of two types. They may be triangular, when the fruit will look like this, triangular, or it may be elongated. It may be elongated when it will look like this, elongated, or sometimes you can tell also oval. Elongated, or you can tell oval, so it will look like this. So a cross was made between a pure triangular type and a pure elongated type, and for this usually this is regulated by two genes. One is T and the other is D. That means when capital T, the other may be capital T or small t, and both these are D, it will give rise to triangular. And when both are small t, then the other one is capital T. This may be capital T or small d, then also it will give rise to triangular. Then if it is capital T, the other may be capital or small t. The other one, this one also may be capital D or small d. This also will give rise to triangular. And when all are in the lower case, and then it will give rise to oval or elongated. So when a cross was made between a pure triangular type and a pure oval type, triangular with a oval type. So this pure triangular is capital T, capital D, capital D, capital D, and the pure oval is small t, small t, small d. They are pure types, homozygous types. So here this F1 hybrid will be capital T small t, capital D small t. This is the F1 hybrid. And when both capital T and capital D are present, so naturally it is triangular. Triangular. Now, when the F2 generation is done by selfing the two types of plants, that means capital T, small t, capital D, small d, and it will be crossed with the similar type. Then the four types of gametes will be produced both in the male and female side. So in this side it will be capital T, capital D, capital T, small d, then small t, capital D, and small t, small t. So in this side also it will be capital D, small t, capital D, small d, small t, capital D, and small t, small d. So in the checkerboard, what is happening? You see, in the Punnett square there will be 16 plants. Now see, in the last case, here it will be small t, small t, small d, small d. When it will be oval. That means see, this one is oval. And in all other cases, here you see both are dominant. That means it will be triangular. And here, since here capital T, capital D are there, that means all these four cases will be triangular. Here capital T, capital D are there, this means all this will be triangular. Easily you can remember. So this is 4, this is 3, 7. Then here capital T is there, here the capital D is there. Then here capital D, here capital T is there. So in these 9 cases, both dominant are present, so they will be triangular. Again, when only capital T is there, so only capital T is there, which case? This is capital T, small t, small t. So this will also be triangular. This case also capital T is there, small t is there, small d there. So this is also will be triangular. And here also capital T, small d, small d, this will be triangular. So are these 3 cases capital T is there. Now in these remaining 3 cases capital D is there. So they will also be small t, capital D, and in this case small t, capital D, small d, and in this case small t, capital D, small t. So they will also be triangular. Because the behavior of the both capital T and capital D is the same, that means they produce the same character. That means we are getting 15 triangular and 1 oval or elongated. Why? Because the T produces one character triangular, D also produces the same character triangular, and when both are present together, they also produce the same. That means in case of the Mendel's dihybrid ratio 9:3:1, that means this produces triangular, this produces triangular, and when both present also triangular. That means all these are triangular. Only this one where double recessive present, that is oval. So this is one of the very good examples of duplicate genes. We can discuss also one second example, that is owned and unless arise. So that we will discuss briefly for the purpose of example. So the example number 2, this is owned and ownless rice. Now what is owned and what is ownless? In some rice you will find one spike-like structure is present like this. This is owned, and if it is not present, then it is ownless. So this character is also regulated, this owned character is regulated by two different genes present in separate loci, but their behavior is same. So here, suppose we will take A1. So this will give rise to owned. We will take A2 gene, or that the other one may be both homozygous or heterozygous. It also gives rise to owned character. And if both A1 and A2 are present, it will also give rise to owned character. Only when all are present in the heterozygous form, all are present in case of homozygous recessive form, then only it will be ownless. So if a cross is made between a pure owned with a pure ownless, this is a ownless and this is owned. So the F1 hybrid will be capital A1, small a1, capital A2 and small a2. So here both A1 and A2 are there, so this will also be owned. And when we will make the Punnett square for the F2 generation, the similar type of ratio will get. We will not draw all those things, so it will be unnecessary, it will take time. So only in this last one we will get the recessive A1, A1, A2, A2, which will give the ownless, and in the remaining 15 cases as we have shown here, it will be owned, because either both the dominant genes will be present in 9 cases and one dominant gene in 3 cases, the other dominant in 3 cases. So that 9:3:3:1, that means these 3 types of ratios will be clubbed together into 15, and this is 1. So these are the 2 examples of duplicate genes or pseudo-alleles. So duplicate type of gene interaction is a non-allelic interaction when 2 independent genes present in different loci, but their behavior is same. They produce the same phenotype when they are present either individually or both are present together. So this is all about the duplicate genes or pseudo-alleles. I hope you have clear understanding. Thank you very much. God bless you. Have a good day.